Okay. Okay, so there are kind of certain interesting topics in cancer biology, and then there are topics which people like I find interesting, and then there are like generally interesting topics. So like metastasis, that's a generally interesting topic for most people. Angiogenesis is another generally interesting topic. So I've picked the topics which I think are most generally interesting. So today we'll look at angiogenesis, but you can't really talk about angiogenesis unless you talk about a phenomena called heterotypic signaling. So we've got to really kind of plow through a bunch of heterotypic signaling before we can talk about the actual process of angiogenesis, which is new blood vessel formation as part of tumor genesis, as far as we're concerned. So when we look at heterotypic signaling, we're going to look at the role of non-cancerous cells in tumors. Because when we look at papers and things, we're always talking about, the cells, and we say they're cancer cells, and we talk about the tumors. And the implication there when we talk about a tumor is that the whole thing is cancerous. That if we took the tumor and we dissociated it, and we took those pieces and we put them into a mouse that would allow us to do that kind of transplant experiment, the implication there is that all of the pieces of the tumor would be tumorigenic, and that's actually not true. There are lots of parts of tumors which play support roles in supporting the growth of the cancerous cells. And so this heterotypic signaling that I'm talking about is the conversations that occur between the non-cancerous cells, and we're going to talk about stromal cells primarily here. So these are cells like associated with connective tissue. So we're talking primarily about carcinomas and adenocarcinomas, and we'll talk about the connective tissue associated with those. So this is loose connective tissue. If you've taken anatomy and physiology, this is loose connective tissue. It's known as stroma. We're talking about things like macrophages, fibroblasts, adipocytes. We're going to focus on macrophages and other immune cells and macrophages. So we're looking at the conversations which occur between these kind of dissimilar cell types. So epithelial cells being significantly dissimilar from macrophages, monocytes, mast cells, and those kinds of cells that you find in stroma. So that's what heterotypic signaling is. It's complicated conversations involving signaling molecules between cells which are very, very dissimilar. Now you do see this in normal biology. So this isn't a tumor-specific phenomena, but you see it significantly more in tumors than you see in normal physiology. And there's a place in normal physiology you see it a lot, and we'll talk about that as a comparison with what's going on in tumor biology. So this is actually a tree, which I thought if I played with it, I could make it look like blood vessels, which I thought I did a pretty good job of. So it's actually a tree. It's kind of cool. Okay, so go back, like way back to the beginning of the semester, and we talked about just kind of a big overview of cancer, if you like. And we talked about that cancer as a disease is really a disease of cells, but it manifests at the level of tissues. You see tissues being disrupted, and then you see organs being disrupted, and that's when you start to see the manifestation of the disease. So many of the cells in a tissue that are non-cancerous end up being manipulated by the cancerous cells. And so that's where this heterotypic signaling comes in. So there's going to be the manipulation of cells that are not cancerous in a tissue. And it turns out that that manipulation by the cancer cell is so that these non-cancerous cells provide support and an appropriate, a suitable growth environment for the cancerous cells. So there's a ton of cells in the tumor which provide this support function, which are being manipulated by the tumor via these signaling pathways. Is what you kind of want to take away from this in terms of big picture is that not all the cells in a tumorous mass are tumorigenic. In fact, that's probably the minority of cells in many, many cases. So if we look at a couple of tumors here, this is an invasive ductal carcinoma from breast, and the membranes of the cancer cells have been stained brown. So you just about make out that the kind of duct is around here. And so the tumor's in here in the duct, and this is an aggressive invasive ductal carcinoma. The cancerous cells are outlined in blue and brown, and the blue cells, those are stromal tissue. So you can already see that a large amount of the tumorous mass here is not cancerous cells. There are these other cells which are doing something in the tumor, and it turns out they're being manipulated by the cancer cells. On On the right is a colon cancer, and so the cancer is on this side, and these are the crypt-like structures, or the kind of like ductal structures that you get in colonic epithelia in the tumor over here on the right, and over here on the left. This is ostensibly normal tissue, so this looks relatively normal. Now the brown dots here are fibroblasts. And so the fibroblasts have been stained brown. And so what you see here is that in the normal tissue, yeah, there are fibroblasts, there are brown dots, but the tumor-associated stromal tissue has recruited huge numbers of fibroblasts. So the fibroblasts have been recruited there for some purpose. And we'll look at what those fibroblasts do and the signaling that they get involved in. But this is not an uncommon phenomenon where you see fibroblasts, macrophages, other types of immune cells recruited into the stromal tissue of the tumor to provide some set of conditions which are favorable for the growth of the actual cancerous cells. So that's one example. Here's a really, really extreme example. This is a lymphoma. This is Hodgkin's lymphoma. There's two kinds of this. There's non-Hodgkin's and Hodgkin's. So these are all lymphocytes, and they're all normal. This is a tumorous mass. These are all normal tissues. This is the one single tumorous or carcinogenic cancerous cell in the image. It's got a Reed-Sternberg cell. And so that's the one cancerous cell. And the rest of the tumor mass is made up of these completely normal lymphocytes. So the mass, if you took these cells and you injected them into a nude mouse, they wouldn't form a tumor. A nude mouse, we'll talk about nude mice quite a bit today. A nude mouse is a mouse which lacks an immune system, and so it doesn't reject xenografted material. So you can transplant cells, you can transplant tissues, you can do xenografts and things, and the mouse won't reject it. So you'll read about two types of nude mice. Nude mice, and then what's called a RAG1, RAG2 mouse. And as far as you can tell, they do the same thing, that they can be used to do the transplant experiments. When you guys have seen these experiments in the research papers where they inject cells into a mouse, those are often nude mice. They have to be kept under very, very sterile conditions, otherwise they get sick and just die of getting infections and things. So they're kept in very, very sterile conditions in labs. So that's a really extreme example of how a mass of a tumor can be primarily non-cancerous cells. So the types of cells that we're going to see that are going to be composed, are going to be kind of making up the non-cancerous parts, so the stromal part of tumors. If we're talking about adenocarcinomas and carcinomas, cancers of epithelial tissues, that's what we'll focus on primarily. Things like fibroblasts, and then fibroblasts can actually differentiate into myofibroblasts. Those are cells which have contractile properties, so they're like they have myofibrils, so they can actually contract, and we'll come back to those later. Endothelial cells, you guys know what those are? They're the cells which basically end up making the lining of blood vessels, of vasculature. And then parasites are cells which kind of wrap around the outside of endothelial cells on vasculature. So those are going to become important when we talk about angiogenesis. A little bit about smooth muscle cells, but not as much, only really in the context of myofibroblasts. Adipocytes, no, we won't talk about those. We'll talk a lot about macrophages and certain types of lymphocytes, and then mast cells a little bit towards the end. But these are all the kinds of cells that you're going to find infiltrating into the tumorous mass that themselves may not be tumorigenic at all. You take them out and put them in a nude mouse, and nothing happens. So those are the kinds of things that we'll look at. And so if you take a tumor and you resect a tumor, so you cut it out, and then you dissociate the tumorous mass into single-cell populations. So you treat it with enzymes which break down the extracellular matrix and break down all the cell-cell junctions, and you end up with single-cell populations. You know how we talk about flow cytometry, and you can use certain cell surface markers to separate cell populations out? So you do that, and you separate out all the CD45 cells. And so those are leukocytes, basically. So they're a white blood cell population. You take the tumor and you separate all the leukocytes. Is what you can see is that if you compare normal tissue distributions of the different subpopulations of leukocytes, so you take leukocytes and you look, and there are lots of different populations based on all these immunological markers. We're not worried about what these things actually are, but you probably recognize CD4 if you've talked about HIV ever. If you look at normal tissue, you see this kind of distribution of these types of basically white blood cells. And then if you look at a tumorous tissue, you see complete shifts. So that's the CD3, CD4 T cells in the normal tissue that's next to the tumor. You can see the tumor has either converted white blood cells that are in place, or it's recruited these white blood cells to the tumor. Now, you're probably sitting there thinking, well, why would a tumor recruit immune cells? That's a stupid thing to do. But one of the things that immune cells do, besides fight off things which aren't going to be there, is they release a lot of different types of growth factors. And so that's kind of the connection we'll come back to making. You see some of them going up. You see so the blue population increased, the green population decreased. And so you see this complete shift in terms of the types of populations of T cells in the tumor if you dissociate the tumor. Okay, so the point here is the cancerous cell is manipulating the local environment to make the local environment better. And better could mean more nutritive. It could mean it's recruiting cells which are going to break down certain tissue structures to release growth factors. It could be any number of things that is motivating the change in the local tumor environment. It's not just a mass of cancerous cells. That's the thing you've got to kind of dispel, because that's what people bring in, is the idea that a cancer is a mass of cancerous cells. It's not the case, right? In fact, there are models which say that the minority of those cells in a tumor are cancerous, and they're often called cancer stem cells, but we're not going to get to that. So we see the shifts in cell populations, and that's just one example looking at immune cells. So what we have is we have the cancer cells signaling to all these different populations of cells that we see, that we could find in tumors, and in turn these are signaling back. This is the heterotypic signaling. This is the signaling between fundamentally different types of cells. In a carcinoma or adenocarcinoma, that's an epithelial cancer, these cells, epithelial cells, keratinocytes, those kinds of things, very, very different structurally and functionally from endothelial cells, myofibroblasts and fibroblasts. These are contractile, these are crawly, these shouldn't be doing any of the things that these things are doing. And so although there are certainly cases in biology where different cell types signal to one another, that's normal. The signaling that we're talking about here is vastly different than you see elsewhere, with a few exceptions, and we'll look at that exception. So all of these interactions are important in the growth of the tumor, and what we've got to do ultimately is we've got to get the tumor from being 1 to 2 millimeters in diameter to much bigger. And the critical step in doing that is going to be angiogenesis. You can't get above 1 millimeter or 2 millimeters unless you actually get to the point you can recruit blood vessels. So some people are going to say things like the whole manipulation that's going on here is so that you can get what's called an angiogenic switch to trigger. I think there are other things going on before you get to that point. For example, some of this signaling that's going on, particularly with cancer cells and fibroblasts and cancer cells and macrophages, is linked to an epithelial to mesenchymal transition. And we'll talk about that again. We talked about it before. So all of these interactions are important, not only in tumor initiation and getting the tumor growing, but getting the tumor able to grow beyond a certain size. So if we've got cancer cells, epithelial cells, they're going to be in one part of the tissue, right? And then the underlying connective tissue is in another part. I'm using tissue loosely here. If we think about skin, for example, you've got the epithelial cells, and then you have a basement membrane, and then below the basement you have the connective tissue, and then adipose tissue and that kind of stuff. Separating the epithelial cells, which are going to become the cancerous cells, from the connective tissue, we have basement membranes. And we've talked about the basement membrane before as a kind of a barrier to metastasis. It's not a barrier to signaling, though. So the signaling that you get from the epithelial cells, the cancerous cells, to the non-cancerous cells, that signaling can pass straight through the basement membrane. It's this somewhat densely packed matrix of protein and carbohydrate, and that forms the basement membrane that separates the epithelial cells from the stromal material, the connective tissue below. That barrier is completely permeable to certain signaling molecules and is actually involved in trapping other signaling molecules. So this is a form of extracellular matrix. So certain things are allowed to pass through. If other signaling molecules have structures which cause them to bind to any of these components here, then they will get trapped in there. So certain growth factors will be trapped, and certain growth factors will be able to pass. Now, the trapping thing sounds, well, that's a good thing because we can interfere with the heterotypic signaling. We can perturb the signaling from the cancer cells to the non-cancerous cells, and we could maybe stop that recruitment in. But the fact that certain growth factors get trapped in here means that at some point they can be liberated later. So extracellular matrix that you find in tissue is a reserve, is a store for growth factors. And if you think back to some discussions we had a few weeks ago, we talked about how bone is a very nice place for cancer cells to migrate to. Breast cancer cells often go to bone. Bone is a nice place to go. What is bone primarily made of once we get past the calcium and phosphate and stuff? What kind of tissue is bone, effectively? It's primarily connective tissue. Okay, if you watch any of those videos that were in the PowerPoint, and any of those links, is what you saw. There was a ton of deposition of extracellular matrix. And in that extracellular matrix, you trap growth factors. So as soon as you start breaking that growth, that matrix down through the act of, for example, osteoclasts, you suddenly start releasing all these growth factors. And those then become involved in the signaling. They should be trapped. They shouldn't be going anywhere. If they're getting produced and they end up getting trapped, that's for a reason. So you can show this happens, and you can show that it's important in angiogenesis as well. Endothelial cells, so here's a tube of endothelial cells making what would be a capillary or some blood vessel. Endothelial cells release a protein called platelet-derived growth factor. I'm going to use a lot of growth factor names, and you don't need to memorize them. I'm not going to ask you what does scatter factor do on the exam or anything. They're just their names so that we can have the discussion, basically. Platelet-derived growth factor normally is meant to get trapped in the extracellular matrix, which is around endothelial tubes. And when it gets trapped there, the point of that trapping is that it stays localized. So the extracellular matrix, which is deposited by the endothelial cells around the endothelial tube that's forming, traps the platelet-derived growth factor. And the signal then recruits to the extracellular matrix and therefore to the endothelial tube parasites, or precursors to parasites, which then wrap around the endothelial tube and drive vascularization. So the trapping there is actually having a purpose. You don't want it diffusing away into the tissue and getting into blood supply and going elsewhere. You want it trapped. And if you mutate the platelet-derived growth factor, and you can make these endothelial tubes form in dishes in labs. So you can create them in a way that they produce a mutant PDGF, so that it doesn't bind to the matrix, to the extracellular matrix produced by the endothelial cells, and therefore it just diffuses through it, and the signal doesn't get localized to the endothelial tubes, and the parasites never get replaced. So you can demonstrate that in some cases you want the growth factors to fuse and get away and act as distant, in a paracrine sense, somewhat distantly. And you can also demonstrate that in other contexts, the localization of the growth factor to a particular site by trapping it in extracellular matrix is important in that context. So unfortunately, there's no simple generalization we can make here. These different growth factors work in different ways based on the context of the tissue and the nature of the heterotypic signaling that's going on. This is what makes signaling so complicated, is it's very tissue dependent, it's very cell dependent, and it's very dependent on the type of signaling we're talking about. You're taking this 4,000 level cell signaling course? We have a 4,000 level cell signaling course. It must be the hardest course in the world to teach, because it's just, here's another example of how signaling works, and there are just thousands and thousands of different examples. Very hard to come up with generalizations. So, we've got all this heterotypic signaling going on, and it turns out that when we get a tumor that starts to form, the tumors that form, their structure ends up being completely dependent on the stroma and the extracellular matrix that develops with the tumor. So it's not just the cancerous cells that form a cancerous structure. The tumors architecture and structure is dependent on the stroma, so the tissue, and as part of that tissue, it's dependent on the extracellular matrix. If you take the tissue context away from cells, you don't get the normal kind of tumor architectures that you see. Now, we've talked about before how if you take colon cancer cells and they metastasize to the liver, that you get colon-like structures in the liver, yeah? And if you take breast cancer cells and you metastasize them to somewhere else, you'll get ductal structures or you get lobular structures. That is completely dependent on having a stromal architecture at the place where the metastatic cells go. So here is a prostate tumor. I'll come back to this little tiny weeny table over here. Here is a prostate tumor. Here's a colon tumor. These are surgical specimens. Now, if you blow these up, you're going to see that this is ductal. Prostates are secretory glands, so they're duct-like structures. These little kind of holes, those are ducts, and these are neoplasias, so these are probably ductal carcinomas in situ, so the ducts are starting to fill up with the epithelial cells. And you can again see that whatever that duct-like structure is there, or that crypt-like structure, is starting to fill up with cells. So here are the two primary tumors. If you take that tumor and you graft it into a mouse or something, because you put the graft into a tissue-like structure, you give it stroma, fibroblasts, macrophages in the environment you put the tumor, that you graft the tumor into, you get a tumor which looks like the primary tumor, yep? So you could put that tumor in, you take that tumor from the prostate and stick it in the liver, you're going to get the prostate-like tumor appearing in the liver. Same thing with the colon tumor. Now, if you take that tumor and you make a cell line from it, so what that means is you take the tumor and you dissociate it out into all the cells, and you try and get the cells to grow in the dishes. And is what you're interested in are the cancer cells, not the stromal cells. The problem here is actually that the stromal cells grow really, really well in tissue culture, but the cancer cells don't. And that supports the idea that cancer cells must have the stromal support. But if you separate that tissue out and you just have the cancer cells, they are really, really bad at growing in dishes. You would think they're cancer cells, so they're going to grow fantastically well in dishes. There are only several dozen cancer cell lines that have been developed. It is really hard to get cancer cells from a tissue to grow in a dish. And it's because they don't have a tissue architecture. So if you can get the cancerous cells in this tumor to grow in a dish, you can make some cell lines. This is called a PC3M cell line. I don't know what the cell line is. If you take the cancerous cells growing in a dish and you put them into a mouse, you get nothing that looks like the original tumor. What's happened is that when you grow the cancer cells in the dish, they go beyond their normal cancerous state. They acquire a number of additional genetic changes, which means they can now grow in dishes, but they don't form structures which look anything like the actual tumor structure. So the point of this whole discussion is that the tumors must have a stroma environment to form tumor-like structures. They're completely dependent on that. And the good thing is, if you can interfere with that, that's potentially therapeutic, right? If you can mess up a cell's ability to interact with its stroma environment, it can no longer really be a cancer cell. It's got to do something else at that point. I mean, it could do something worse, I guess. So, okay. Now, I said that the extent of heterotypic signaling that you see between the stromal cells and the cancer cells is generally not the same to the same extent that you see in normal biology. And I said there was an exception to that. Turns out that in the mid-1980s, whenever it was, somebody, I don't know who it was, somebody noticed that you see the same type of heterotypic signaling in cancers, in tumors, that you do in the process of wound healing. Now, before this was observed, this was only 30 years ago, before someone noticed this, people didn't know whether the tumors were reinventing the wheel. Were they coming up with new ways to interact with macrophages and fibroblasts and myofibroblasts and mast cells and all those kinds of things? Were they building this heterotypic signaling relationships? Were they building them from scratch every time? So every time a tumor arose, did it have to go through the process of evolution to generate these interactions so that it could recruit and develop the structure it needed? Or was it manipulating some existing pathway? And when this person, whoever it was, recognized that there was the same kind of heterotypic signaling and the same type of physiological processes going on in tumor genesis as there were in wound healing, it pushed us to the point of thinking, this is just the cancer cell manipulating an existing physiological set of pathways and manipulating an existing set of heterotypic signaling relationships. So, you remember how we had to begin the semester with this phrase? You guys remember the phrase I'm talking about? That it's the inherent regenerative morphogenic potential of metazoan cells which underlies the potential for cancer or something like that, yeah? Here is the second really nice example of how cancer cells don't reinvent every single time the cancer appears. So one of the things you'll get on your final is you'll get the question you've had before, but you've got to do a way better job of answering it because now you've got examples from metastasis, I'll give you an example from angiogenesis with wound healing. So we've got to talk about wound healing a little bit, and then I'll show you the relationships between tumor genesis and wound healing. So we'll talk about wound healing a little bit, which you may not have seen before. Easiest way to think about this is you cut your arm, you break skin, basically. You skin a knee or something like that. So that is ultimately what's going to happen. First thing that happens when you cut yourself, what do? You bleed. So you damage vasculature. So you damage vasculature, and then basically stuff leaks out of your blood vessels, and one of those things are things like thrombin and fibrinogen. And that gets into the tissue, and fibrinogen converts to fibrin, and that starts a clot, and platelets get trapped in there, and that's the next thing that happens, yeah? Unless you're hemophiliac, then you don't stop bleeding, but here, you blood clots, and so you get a clot at the site of the injury. So there's also, that's that part there, and that's what leads to forming the clot. So that's the fibrinogen to fibrin cascade, and that's platelet aggregation. This is actually one of few examples of positive feedback in biology, actually, blood clotting. Once you start it, you don't want it to stop. You know the other ones, yeah? Yeah, the other famous one? Childbirth, yeah. I've not had children, but I'm pretty sure that once that starts, you don't want it stopping and then starting and then stopping. You want it to go to completion, I'd imagine. Platelets also release a lot of signaling factors. There's PDGF we talked about before. TGF-beta, that's going to be one of the ones that it's useful just to have that as knowing that it's important, not necessarily knowing every single thing it does, because it's a really complicated growth factor and does different things in different contexts. But it's along with PDGF and transforming growth factor beta, those are two that are important growth factors. My cell signaling class in college, we spent the whole semester on 3 growth factors: PDGF, transforming growth factor, and scatter factor. So these 2 growth factors, platelet-derived growth factor recruits fibroblasts, and fibroblasts end up, some of them become myofibroblasts. So you've at least seen those being involved in stroma. Myofibroblasts can release certain growth factors, don't worry what they are, and they can recruit endothelial cells, and that can drive angiogenesis. Now you've got to do the angiogenesis thing when you break vasculature, because you've got to replace the damaged vasculature, yeah? So when you make a cut and you damage blood vessels, you rebuild, you don't repair those blood vessels, you build new blood vessels. Basically the ones that get damaged, get broken down and degraded, you replace. You put new blood vessels in place. So part of this whole damage event up here, the whole wound up here, part of healing has to come down through angiogenesis. So as well as recruiting fibroblasts, we recruit macrophages. What are macrophages going to do in a wound? One of them. When you think about macrophages coming into a wound, why are they coming in? Yeah, they're to clean the wound up, to prevent, to eliminate anything that was on the rusty nail that you scraped yourself on. So macrophages get recruited. So you're familiar with that happening. But it's not just an immune reaction. It's not just recruitment of a cell type to eliminate potential pathogens and to clean the wound up. Macrophages do a bunch of other stuff as well. They release a group of proteins called matrix metalloproteinases. Those are proteases. They break down extracellular matrix. And in so doing, they release growth factors. So we talked about growth factors being trapped in extracellular matrix. If you're going to grow new vasculature, you need space in which to do it, right? So you actually have to start breaking down the tissue a little bit to create space. You need the physical space for the endothelial cells to start forming tubes. And so the matrix metalloproteinases basically will end up breaking down the extracellular matrix. That has two effects. Releases growth factors. Cytokines are growth factors which drive cell division. You need that to replace damaged and lost cells. And you're also creating new spaces for the endothelial cells to form. So a couple of things that are happening there. In terms of the overall process, epithelial cells at the edge of the wound basically undergo an EMT. So that's one of the places in normal physiology you see epithelial cells becoming mesenchymal. You have to basically get the edges of the wound to crawl under the wound to close off the opening. Okay, and they crawl under the blood clot and across the top of the stroma. And so you create an EMT, you get proliferation and you get crawling into the wound site, and then you get the reversal of the EMT, you get the MET, which you talked about in that paper. So the cells go from being mesenchymal when they crawl in to cover the wound to going back to being an epithelial cell type. And part of that is cellular proliferation, and you basically reconstructed the epithelium. So this part here, the critical part, is that you've got a lot of signaling between a lot of different cell types. Okay, we've got signaling between platelets and fibroblasts, platelets and macrophages. We've got all kinds of different growth factors that are being released. We've got the recruitment of different cell types, endothelial cells, connective tissue cell types. We've got the recruitment of immune cells, macrophages. So there is a lot of heterotypic signaling going on in this process. And it turns out there are remarkable similarities when you look at the processes that go on during tumor genesis. There are some differences. So this is one of these exam questions. What are the similarities? What are the differences? And then how is this a good example of an existing morphogenetic program being usurped by tumors? Okay, if you want to look at this graphically, here is what happens. Here's the wound site, here's the damaged wound. Okay, the fibrin clot forms, it's the start of the clot. Platelets aggregate. Macrophages get recruited in. Macrophages release different types of TGF, TGF beta and TGF alpha. They basically act on epithelial cells at the site of the wound. So right in here at the side of the wound, these are epithelial cells. TGF beta and TGF alpha basically trigger an EMT. They also stimulate the proliferation of epithelial cells. So you have proliferation of epithelial cells and you have an EMT. And so the EMT causes the epithelial cells to be mesenchymal and they start to crawl. They form these fingers which kind of come down under the tumor. Under the wound. Okay. We've also got the release of growth factors by the macrophages, which end up leading to angiogenesis, so the recruitment of endothelial cells. So one of the growth factors we're going to come back and talk about is a growth factor called VEGF, vascular endothelial growth factor. Macrophages and platelets, the platelets degranulate, so they release a lot of growth factors, and the PDGF released by the macrophages basically can drive the fibroblasts to become myofibroblasts. So what the myofibroblasts do is they help to pull the two edges of the wound together. So they have these contractile properties, and they can actually act like little tiny kind of muscle fibers and start to pull the edges of the wound together. You've probably seen, if you ever cut yourself and it starts to heal, do you ever see the skin slightly pucker? I'm a nightmare because I pick everything off. I'm a picker, so but it's what you'll see is you'd often see the edge of the wound start to pucker, and it's because myofibroblasts are exerting a force on the skin, trying to pull the two edges of the wound together. And then once you get the two edges closed together, then you're going to have a reversal of the EMT, so you have an MET, and so we're going to close the wound over. The myofibroblasts are also really, really important in recruiting endothelial cells. So there are several things here that you see in tumor genesis. Okay, angiogenesis is one of them. There's also the EMT and the MET that we've talked about before. So there are at least two major things, and that's before we even look at the heterotypic signaling that we see going on, and we look at any of the other phenomena. If you look at tumors, like I said, you see these similarities between wound healing and tumor structure. If you look at newly formed vasculature in a wound, it's not fully formed, and so it's somewhat leaky. If you look at healthy, fully formed vasculature, it's not leaky at all. And so you can test the leakiness of vasculature by injecting dyes, and they're carbohydrate dyes. This one's red dextran. And so it's too big. It shouldn't get out of vasculature. Vasculature is slightly leaky anyway because you have to have edema, you have to have fluid that can escape from vasculature. But it shouldn't be really, really leaky. And so this red dextran dye basically shouldn't leak out of normal, healthy vasculature. So in normal vasculature, you should get no leakiness. That's what that looks like. If you look at tumor vasculature, though, it's really leaky. So that's one difference between the vasculature that you get in normal tissues and that forms when you're done with normal wound healing. It's totally fine. But when you look at tumor vasculature, it's crazy. It's so leaky, and that has some consequences for therapeutics. We'll talk a little bit about that as we get towards the end, I think. So there are some differences as well as some similarities. So that's a difference between normal vasculature and tumor vasculature. One of the similarities you see in wound healing and in tumor genesis is if you look at a lot of the stroma that you find in these adenocarcinomas and carcinomas, you find a lot of bundles of fibrin. So that's fibrinogen that's been converted to fibrin. So it looks like it's trying to clot. And so that's to do with the recruitment of certain cell types into the stromal tumor. So what's going to happen in a tumor is that when you've got this kind of tumor vasculature, things like thrombin and fibrinogen can leak out of the vasculature into the tumor. And in the tumor, where you've got cells that are producing the enzymes that aid in the clotting process, you end up with these bundles of fibrin forming. So in this breast cancer, this brown staining are bundles of fibrin. And I don't know what kind of cancer this is, but you can see these fibrin bundles here. So that's a consequence of bad vasculature or leaky vasculature. But it's a similarity that you see between wound healing and tumor genesis. So there's some similarities between those two processes. Why do you care about fibrin being in tumors? One of the things that fibrin does, besides forming a clot, is it forms a matrix which cells can migrate on. So one of the things, once you've got a clot that starts to form, you need to get macrophages and fibroblasts crawling into that. The macrophages obviously need to come in so they can clean up the wound site. They provide a substrate along which cells can crawl. Why would that be beneficial if you're a tumor? It's localized invasion. It's an opportunity for cells to be able to crawl through some kind of matrix. Sorry?
I was just wondering what the mechanism is for why there's so much leakiness in the vasculature. Is it overexpression of things like VEGF1 that mess that process up, or is it just the inherent instability in the genes that make it so that the angiogenesis doesn't happen correctly?
I don't know. The short answer, I could speculate. And the way I would speculate is I would say that if you try and manipulate a process and you try and mimic it, it's like trying to make a rip-off of a high-quality product. When you try and make a rip-off of a high-quality product, you end up with something that's never as good. And so I don't know what's going on exactly here, but one of the things I've always thought here is that when you try and manipulate a signaling pathway, you don't ever get it quite right. The signals that are being produced are the wrong mixture of signals exactly. And so rather than getting this, which is due to the correct abundance of endothelial cells and parasites, I bet you're not recruiting parasites correctly here. And so that's going to be something to do with what PDGF-beta is doing. That's what gives endothelial, that's what gives vasculature its integrity, the parasites. And so if you don't get parasites recruited, you're going to end up with poor integrity. If you look at the vasculature, you can see big holes. The cells don't overlap properly. So we'll look at that at the end. But I think it's just a consequence of the fact that you're trying to manipulate a system and you're not getting it quite right. And so it doesn't happen as well as it should under normal conditions.
Then is it more difficult to recruit and activate lymphocytes because you don't tend to see any lymphatic drainage around those kinds?
I would presume so. Yeah, there's a whole I think there's a whole section in that, whatever this chapter is in the book, 13 or 14, which talks about lymphatics in tumors. And it's one of those things I decided to eliminate, but we can certainly take a look at it more closely. Yeah. But there are weird things that go on with lymphatics as well. Yeah. I don't know what you use to build lymph vessels, to be honest. I've never taken anatomy and physiology, so I don't really know what you need to build lymphatics, but the lymphocytes would be what's in them. So I don't know whether there's something weird about the recruitment of lymphatics. The actual structural cells, the equivalent of endothelial cells, whatever they would be in lymphatic vessels. I don't know. So. Okay, so are you guys seeing that there's this important relationship between cancer cells and the stromal cells, and that relationship can be manipulated? That's the kind of where you need to be right now. Yeah. Okay, to kind of strengthen that idea and give you an idea of how important the stromal cells are. You can drive a cancer in a tissue without any mutation to the cancerous cells themselves, which is completely contradictory and completely flies in the face of a lot of stuff we've talked about. So you can drive the first step in tumor genesis, hyperplasia. You can drive that purely by manipulating the stromal tissue, the connective tissue. So here is a ductal epithelia. I'll have to look at my notes to tell you what it is. It's prostate. So here's normal prostatic epithelia. The stroma is the kind of the white tissue with the odd nuclei stained in it. Those are macrophages, fibroblasts, adipocytes, that kind of stuff. So that's the stroma. Here is the ductal epithelia. So this is prostate, so this is producing semen. So this is normal. In this tissue, there is signaling across the basement membrane, which is around the duct. There is signaling back and forth, a certain amount of heterotypic signaling back and forth between the epithelial cells and the cell types in the stroma. Okay, and that's totally normal. That's the macrophages communicating with the fibroblasts, and any T cells which are in here communicating with one another and with the macrophages, and all the different cell types communicating with the epithelial cells in here. Okay, here is an intraductal neoplasia. They call them pins, prostatic intraepithelial neoplasias. So it's the same tissue. You can see that there's some kind of duct, but it's now filled with cells. There are no mutations. When you look at these genomically, there are no mutations in them. They're doing exactly what they should normally be doing. The only thing that's been done is that the system has been manipulated so that the cells in the stroma no longer have the transforming growth factor beta receptor. So we've taken the receptor out. The TGF-beta receptor has been deleted from the stromal cells. So this is a complicated genetic engineering experiment in a mouse, basically. So you've mutated the TGF-beta receptor. Now, what that receptor does is it normally regulates the signaling from the stromal cells to the epithelial cells. And so normally the stromal cells are sending out a certain amount of signal, whatever those signals may be, and the epithelial cells are responding to that signal. If you take away TGF-beta responsiveness in the stromal cells, they start just pumping out way too much information, way too much growth proliferative information for the epithelial cells. These cells are always communicating with one another in terms of grow, grow, die, die, divide, divide, bigger, smaller, do this, do that. And so the stromal cells have just told the epithelial cells, these normal epithelial cells in the lining here, are just being told to divide. And so you've got an intraepithelial neoplasia and there's nothing wrong with the actual cancerous cells. That's remarkable that you can have a cancer formed due to a single mutation. This is not actually a cancer because it's not metastatic, but it's called a neoplasia. And so this is one of these weird nomenclature things that upsets people. Because when we say neoplasia, people think it's a cancer. So you've got a neoplasia that's formed that's got nothing to do with the actual cells which are forming the neoplasia. That's completely and utterly bonkers if I think about it. If we think about it. because it flies in the face of what we've talked about already, which is you've got to have genetic change in the cells which become tumorigenic, right? Now these cells are obviously dividing too much, so there's a good chance they're gonna start accumulating mutations, but nothing was wrong with them to start with. All we did was make a point mutation in the TGF-beta receptor of the stromal cells. That's the magnitude of the impact that the stromal cells can have through heterotypic signaling. That's how important it is. I think this is one of the most kind of best ways to show how important this relationship is between stromal cells and precancerous and even cancerous cells. So you can show this in lots of ways. Okay, so I'm going to show you several pieces of data, or maybe this is the last one, which shows you the importance of the extracellular matrix and stroma. Stroma is cells embedded in extracellular matrix. So here's the importance of the extracellular matrix and some of those cells. What's been done in this experiment is somebody has taken normal human mammary epithelial cells. So these are the cells which line the ducts. So if we go back to this one, these would be normal mouse epithelial cells from prostate. So what we've done is we've taken the same cell type from breast. So these are human mammary epithelial cells, and they've been transformed. So when we transform cells, we've pushed them down the first, we've made them make the first step towards tumorigenesis. So when you transform a cell, it starts losing responsiveness to inhibitory growth factors. It stops being refractory to apoptotic signals. It stops observing the normal boundaries such as when it touches another cell, it should stop dividing and stop migrating. So that's the first step normally that a cell takes towards tumorigenesis. It becomes transformed. So you can transform them by things like RAS. So you've seen in research papers how we introduce RAS and you get transformation of the cell, and then you inject those cells and you get more tumorigenesis. You can also do things like express telomerase. Telomerase is the enzyme which adds to the ends of the chromosomes. Now you don't have that in most of your cells. You have it a lot during early development, when you have a lot of cell divisions and you need to keep the telomeres long so they don't get really short and stop dividing prematurely. But you can also overexpress this enzyme in cells, and then they'll become transformed. And you can do things like put certain viral proteins in the cell. So any of these mechanisms or combinations of these mechanisms can convert normal human mammary epithelial cells into transformed human mammary epithelial cells. If you take those cells and you inject them into a nude mouse, at day 50, you'll start to get tumorigenesis. So tumors will start to form, and it'll take about 20 days to form a 300 cubic millimeter tumor. That's a pretty small tumor. Okay? And it only happens in about 52% of the mouse you inject them into. So these aren't really, they're tumorigenic, but they're not massively tumorigenic. If you now take the same cells and you mix them with what's called matrigel. Matrigel is a semi-synthetic pseudo extracellular matrix. You can buy it from businesses and things, from biotech companies. And if you want to grow tissues in 3 dimensions and do studies on cells moving in 3 dimensions and forming lobules and ducts and things, you do it in this synthetic matrigel stuff. And so that's like stromal material, but it doesn't have cells in it. If you take the transformed cells, you put them in the matrigel, you've accelerated tumorigenesis by 10 days, and the tumors get bigger. I don't think they form any quicker. I think the rate of change here is the same as the rate of change here. But they get bigger, and you get 100% of the mice that you inject this into getting tumors. If you now take the tumor cells and you add just normal fibroblasts, these are normal fibroblasts, and they're not transformed, they're not cancerous, they're not from a tumor, you halve the time to tumorigenesis relative to the tumor cells plus the matrigel. So you've accelerated tumorigenesis and the tumors get even bigger, still 100% effective. Okay, both of these results here speak to the importance of the extracellular matrix material itself as well as the stroma. So fibroblasts, and you can do this with other cell types as well from stromal tissue. When you've got those present, you're going to get much more aggressive tumorigenesis. People were thinking when they discovered this is fantastic news because it gives us a new therapeutic target. We can start perturbing extracellular matrix formation in tumors. We can start interfering with the heterotypic signaling between between the cancer cells and the supporting cells, the stromal cells. It ended up that it didn't really work out. It's one of those things where it's great, here's a new philosophy and a new strategy, and it's the same as what we saw when we looked at angiogenesis inhibitors in the mid-90s, no, mid-2000s. They never really panned out as therapeutics, but they had huge promise. Okay, so what is going on with the stromal cells in terms of driving tumorigenesis? We're going to see some of the same things that we saw in wound healing with these stromal cells. So we'll start off with the carcinoma cells. You don't have to memorize this, but you should be able to tell it as a story and draw some comparisons with wound healing. So you haven't got to worry about knowing what MCP1 is and what CSF1 is, but you should be able to say there are some signaling molecules which are involved in this process. So carcinoma cells, let's worry about those to start with. They release a lot of factors which attract other cell types from the vasculature. Okay, so those are called chemotactic factors. They're things like scatter factor, which here is called CSF1. PDGF, like we've seen already, is chemoattractive. So that will attract circulating monocytes to becoming tumor-associated monocytes, which then differentiate into macrophages. Macrophages you don't tend to have in your vasculature. They tend to circulate as monocytes, and when they go into a tissue or an organ, then they differentiate. So you'll have lung-associated macrophages. So here, monocytes which end up going to tumors because the cancer cells are pumping out these chemotactic factors are called TAMs, or tumor-associated macrophages. Turns out these are critical. These are probably the critical component, if you've got to remember one of them, in terms of driving this process of support. And subsequently, we're going to be talking about angiogenesis. So the tumor-associated macrophages, one of the things they do, let's do the easy thing first. One of the things they do is they can produce growth factors which support the growth of the cancer cells. So there's a positive feedback loop going on here, right? Okay, so that's one of the things that tumors do. They take a normal feedback loop, which should be self-regulating, and they've now converted it into a positive feedback loop. So we've got a positive feedback loop going on here. Your cancer cells proliferate, they pump out more chemotactic factors, more TAMs. TAMs pump out more growth factors, more carcinoma cells, pumping out more chemotactic. Your positive feedback loop's going on here. Okay, let's go down the right-hand arm. The TAMs also secrete things like VEGF. VEGF is vascular endothelial growth factor. It basically drives the proliferation and recruitment of endothelial cells. And so that is going to lead us down the road of angiogenesis. Okay, these are critical in driving angiogenesis. You have an immune cell driving angiogenesis. Do you see why it's called heterotypic signaling? The immune system is the immune system. Angiogenesis is blood vessel development. Very dissimilar sets of processes, and now they're communicating with one another. That's where the heterotypic signaling becomes important. TAMs also release a whole bunch of proteins which degrade extracellular matrix, and anything you find in extracellular matrix. MMP is the one you want to remember. MMP9, if you're going to remember a specific one, that's the main protein that's involved in these pathways. They disrupt the extracellular matrix, and like we said, you've got trapped growth factors. Trapped growth factors are released. They could be things which drive angiogenesis. They could be things which end up feeding background onto the carcinoma cells. So again, there's another positive feedback loop going on here. Let's talk about IGF-BPs. Remember we talked about apoptosis, and we talked about insulin-like growth factors, and then there were the insulin-like growth factor binding proteins, which blocks the IGFs. Okay, IGFs were one of these growth factors which were pro-survival. So they suppress apoptosis. IGF-BPs, you find them in an extracellular matrix. And if you break those down, you liberate the IGFs, and now you're driving a suppression of apoptosis. So a lot of these things, you can start thinking about them as being kind of working together. So now we've got signaling through apoptotic pathways as well by an immune cell, which is not that far from it, which is closer together than it is with angiogenesis. So we got the TAMs doing pretty much three things. Okay, producing growth factors which drive proliferation in the cancer cells. We've got them releasing growth factors which are pro-angiogenic. and we've got them releasing proteins which are going to be involved in disrupting the extracellular matrix, creating new space, releasing growth factors which are mitogenic, which are chemoattractive. Okay. The TAMs here are the critical, one of the most important cell types in the stroma, which is going to drive tumorigenesis. So hopefully 12 weeks ago, you never would have thought that. When I write, one of my questions I have on the final is often, tell me something you were amazed by that you learned during this class. This is the kind of thing for me, when I learned this stuff, I was like, this is completely nuts. This is kind of, you know, but again, you know, potential therapeutic targets. Maybe we can go after these. What about MMP9 inhibitors? Let's go after those. Whole research programs based around MMP9 inhibitors. Can we inhibit the breakdown of MMP, of the extracellular matrix in an attempt to disrupt angiogenesis and break this positive feedback loop? So the dean of my department when I was doing my undergraduate degree had a whole research program focused on MMPs, looking at this exact thing. He's who I learned cancer biology as an undergraduate from. So we're moving into talking about angiogenesis. We've kind of transitioned from the heterotypic signaling basics through to starting talking more about what's going on in the tumor, and we've started now to look at kind of the angiogenic process. The basic message here is that if a tumor cannot drive angiogenesis, it's never going to get big enough to be a real threat, unless it can become metastatic and seed tumors elsewhere, which become angiogenic. So the problem in the tumor is hypoxia, right? Hypoxia and some other things, but hypoxia is the easy one to start off with. Here are 3 different tumors. I don't know what this tumor is here because it's not legend. It's not in the legend anywhere, but here's a tumor. The endothelial cells, or the vasculature, is shown in green, and staining in red is a protein which is induced by cells that are undergoing hypoxic stress. So this gives you an idea of that's as far as you can get away from a blood vessel in a tumor before you become hypoxic, and then you're going to become necrotic. Okay, you can't do enough work with fermentation alone, and fermentation has other side effects as well, to support tumor growth. You can't make enough ATP to support tumor growth using just fermentation. So here is the problem. Okay, you see it here as well. Here's a melanoma. So the melanoma's the whole thing here. In the middle there, you can just about make out some little brown cells. That's the capillary. So that's a capillary in the melanoma. This is healthy melanoma, if you want to think about it in that sense. Those are healthy cells. When you get less than 0.1 millimeters, and you've got a ruler, look at how big a millimeter is. That's less than a tenth of a millimeter. Once the tumor gets to be bigger than 0.1 millimeters here, all this becomes necrotic. So you see this, and again, the immediate thing is, this is huge therapeutic potential, right? If you can stop the tumor getting bigger than this by stopping angiogenesis, that's massive. Here is prostate neoplasia. And so there's the blood vessel in the middle. Here, just over 0.1 millimeters. And then all this is becoming necrotic and dead. Okay? You can't get above it. Same thing over here. There's the vessel. Maybe a little bit bigger. You can't get bigger than 0.2 millimeters, generally speaking. Now, if you get multiple blood vessels, then yes, you can, and they're closer than 0.2 millimeters. Why is this 0.2 millimeters important? Why are we about 0.1 millimeters? Diffusion of... What's the most important one here? Oxygen. Okay, you're limited here by the diffusibility of oxygen. Okay, oxygen is small, highly mobile, and nonpolar. So it's going to be the most diffusible thing you can probably find in tissues, along with carbon dioxide, smaller than carbon dioxide, so it's going to be better at diffusing in terms of it can diffuse faster. Here's one of my problems with biology classes. We use examples for diffusion, like dropping dye into water. Yeah? You've seen this in textbooks. They drop a piece of dye in the water and it spreads out, and they say that's diffusion. Or they say somebody walks into a room with really strong body odor and you smell them at the back of the room. That's not diffusion. What is that? That's convection. Okay, that is water currents. Even in that little beaker of water, there are currents flowing and things moving. Okay, those examples you see in your ear, they're not diffusion, they're convection. Diffusion works over unbelievably small distances in biological systems. So these gross examples we give you, these macroscopic examples are useless. It's the same as entropy. Have you seen the pictures in books with entropy where you've got a tidy bedroom and a messy bedroom, and they're like, that's entropy. It's like, that's not entropy. There's nothing to do with entropy. Entropy works on microscopic levels. That's not what it's about. So those macroscopic things are misleading. Diffusion works over these very, very small distances. From 0.2 millimeters, 0.1 to 0.2 millimeters is what we're limited by here. You can't get bigger than 0.2 millimeters, or you can't get further than 0.2 millimeters from a blood supply. So 0.2 in each direction, you're looking at a diameter of 0.4 millimeters, half a millimeter. So that's kind of your limit of growth. Limit angiogenesis, you limit tumor growth. You must do, because the tumor is going to self-destruct because it can't get blood supplies. So there are other things as well. Complicated graph. Left side is oxygen partial pressure, right side is pH. And is what we can think about here is what happens to different variables. Okay, glucose concentration. Here's one blood vessel. Here's a second blood vessel. Okay, so there are two blood vessels here on each extreme end of the graph. So imagine there's a blood vessel drawn over here and then another one over here. Okay, and in the middle you get this anoxic zone. Okay, so hypoxia is low, less than 2.5 millimeters of mercury, and anoxia is way, way lower than that. So the oxygen thing is one thing. Oxygen tension dropped way down, whichever color line that is. ATP concentrations drop way down. ATP concentrations drop way down because what happens is that you see an increase in lactic acid concentration. Why is there an increase in lactic acid concentration in these anoxic areas? They do glycolysis and then they ferment pyruvate into lactic acid. And they stop producing high concentrations of ATP, so the ATP drops down, the oxygen drops down, the lactic acid goes up, and the pH drops as well. So this microenvironment is not the microenvironment you want a cell to be able to grow in. Okay, so cancer cells, if they're growing under these conditions, they're going to be burning through glucose at alarming rates because 1 molecule of glucose versus cellular respiration, 34 ATP. 1 molecule of glucose through glycolysis and fermentation, 2 ATP. You need 18 times as much ATP if you're doing fermentation to produce the same amount of ATP to do the same growth compared to doing what you're doing with cellular respiration. So you've got to recruit vasculature. If you're going to be a tumor and you're going to be successful, you've got to recruit vasculature. You can visualize this happening as a process. So what these people have done, this is a neat experiment. You can do this in mice and chickens, and I've seen it in mice and chickens. So you take a mouse and you subcutaneously inject a population of cancer cells. This is colorectal cancer cells. And then you create a small window in the skin and you put like a little glass or silicone, like a cover slip basically, on the skin. And so you have a little window. And then you can watch the tumor genesis and you can watch the angiogenesis. And so you watch it over the course of 3 weeks or so, and you can see increased vascularization of the tumor as the tumor develops. So not only is the tumor developing, and it's dependent on the stromal tissue for growth and support, it's also dependent on the stromal tissue for recruitment of the vasculature. You can't do this in vitro because you cannot get the three-dimensional stromal structures that you get when you do it in a mouse. And now I've said a few times, this is great because this is potentially a therapeutic target. And sure enough, if you look at a tumor that's been grown under conditions in a mouse where you don't interfere with angiogenesis, you get nice angiogenesis and you get good vascularization of the tumor. If you co-treat the mouse as it's developing the tumor with an inhibitor of VEGF, the VEGF receptor, so you get an inhibitor of the VEGF receptor, you can seriously disrupt angiogenesis. And when you look at these tumors, they are on average smaller and don't weigh as much. So this was fantastic. People thought that you could use these VEGF inhibitors. And I remember the one point in my PhD where I was super, when you start a PhD, you start off super excited, and then about a year in, you're like, oh my God, nothing ever works. And it just gets really frustrating. And I went to a conference about 2 years in when I really kind of hit a low, and I was like, nothing worked for like the last year. And I went to a talk by a guy called Judah Volkmann. I don't know what he's doing right now, but he was the grandfather of angiogenesis inhibitors. And he gave a talk at this meeting, and it was a private meeting just for our research institute. And he gave this meeting, and it's the first time in a conference meeting when I collectively heard a bunch of very famous scientists gasp when he showed data in mice about how effective these drugs were at reducing angiogenesis. It was the first time he was showing the data, so it wasn't published. I was sitting with a room of, my PhD supervisor won the Nobel Prize, so he was there, another Nobel Prize laureate was there. These were some of the best cancer research minds in Europe and in the world. And there was a collective gasp. And it was one of those times in your career where you're kind of like, this is awesome. I'm involved in something which is awesome. The feeling in the field in the mid-2000s were that these were going to cure cancer. These were the drugs that we'd been looking for. And the good thing about these drugs is they don't act on the tumor, do they? They act on the normal vasculature. What is the benefit of that?
There's no potential for a workaround.
There's no selective pressure on the tumor. Because you're not selecting the tumor, you're targeting the support structure, which isn't tumorigenic. So you kill the vasculature. The vasculature's got no mode of, the vasculature's not undergoing these rapid genetic changes, so mutations that allow you to bypass the need for vascularization can't develop because you're not targeting the tumor, you're targeting the vasculature. And what's the other benefit of targeting vasculature? Who gets cancer? Adults, right? Generally speaking, exceptions, yes, but most cancers occur in adults. What is not happening in fully formed adult humans? Angiogenesis. When does that happen? It happens during certain stages of development. Yes, wound healing. Yes, you wouldn't want to give this drug. You'd probably have worries if you had someone on these drugs who had a major trauma, because then they couldn't heal. But the other great thing about these were that it's not a normal process in adults, unless there are exceptions. So, let's keep looking at vasculature. These are angiograms. This is kind of a bit of a game. These are angiograms. Do you know what an angiogram is? It's a way of tracking blood vessels. And you can image the blood vessels. What's that an angiogram of? Tell them out. So, yeah, not bad. It's a brain. It's a brain angiogram. What are those then? Those are your eyeballs. What's this one? An angiogram of? It's annoying. It's weird, isn't it? And you're like, I don't know. What structures do I have in my head? And everyone's thinking really complicated stuff, so they're eyeballs. Okay, what's this an angiogram of? I'm often told that I don't have one of these. There you go. I'm often told that I have a small piece of coal in its place. That's an angiogram, that's a cardiac angiogram. That one? This one you can get by looking closely at it. That's an eyeball, those are eyelashes. That's an eye, and that's going to be the optic nerve in the back there with the really biggest blood supply. But that's an eye. So, this one looks a little bit chaotic, right? These are all highly organized vascular structures. They must be because the structure they feed and they support is highly organized. And so normal vasculature is highly organized, okay? This is the vasculature around, this is a tumor, and the way it's been imaged is it appears as this big black hole. This is tumor vasculature. So this is vasculature that's been recruited. This is the edge. This is looking like slightly normal tissue up here, and the vasculature is much more organized. This is normal vasculature. This is tumor vasculature. Goes back to your question, Sterling. I think when you try and mimic something and you don't do a good job of it, it sucks. It's like a prana ripoff, basically. It's not quite right. I think that's what's going on with these things, is that it's kind of like, we don't care whether it's organized. We care whether it brings oxygen and nutrients and takes away wastes. So tumor vasculature is highly disorganized. It's one of the differences if you've got two categories of similarities between tumor genesis and wound healing, or different types of vasculature. Totally disorganized in tumors. So another difference. Normal vasculature is not leaky, like we said before. When you look at tumor vasculature, you get all of these little holes in it. And so these are endothelial cells, and they're not overlapping. They should overlap, should be completely overlapping, so that you'll get some leakiness, and you can induce leakiness, so you can have edema, and you can have extravasation of cells into tissues and fluid into tissues when you need edema and you need plasma and you need thrombin and fibrinogen and you need those things in tissues for clotting. You can induce that in normal vasculature, but in tumor vasculature, you get these holes. This is a magnification just so you can see a little bit clearer. You have all these micro holes and then these bigger holes where you can see the edge of the endothelial cell. It's not overlapping. They're not overlapping entirely. Again, I think it's like you try to build a house, but you build it out of twigs rather than out of brick. You get a crappy house. I think it's kind of, we tried to do it, but we didn't really do a very good job. So, right, nearly finished. What are we on? How many do I have left? 3 left. Okay. Okay, so just to recap this slide, because it's these macrophages which are critically important in driving angiogenesis, and we'll finish up with that as the last point again in a moment. Okay, so these tumor-associated macrophages, they're recruited by the carcinoma cells through these growth factors, and then they do these 3 things. They stimulate carcinoma cells to replicate, they drive angiogenesis by releasing angiogenic factors like VEGF, they secrete matrix metalloproteinases that degrade the extracellular matrix to make space, and they also are going to be releasing growth factors. Okay, so we've said that before, but it's important to reiterate because that's the mechanism by which you're going to get a tumor. I've always thought that if you can mess around with this piece here, I'm thinking about antibodies which block scatter factor and these other growth factors. If you can have antibodies which bound to those growth factors, specifically in tumors, you could stop this recruitment of the tumor-associated macrophages. So I always thought that would be a neat chemotherapeutic. So this has all been really well studied, and that whole tumor-associated macrophage thing has really been worked out in mouse models, where you can make cancer models, and then you can interfere with the signaling molecules by making knockouts and knock-ins and conditional knock-ins and mutating the growth factors and things. So the most popular mouse that this has been worked on is in a mouse which you can induce pancreatic cancer in. And so you can induce tumor genesis, and what you do is you induce hyperplasia in the islets or Langerhans, and so those are the insulin-producing cells in the pancreas. So you can trigger the mouse so that it produces hyperplastic islets, and then you leave it alone, and you can study what causes it to go into this angiogenic state. And so once the hyperplastic islets start to recruit vasculature, then you get full-on tumor genesis. So you get this transition from hyperplasia to the more aggressive stages of cancer that we've talked about. And so you can manipulate the mice, and you can cross the mice with other mice, and you can knock out certain growth factors, and you can knock out certain receptors for growth factors. And it turns out that what's happening is that the hyperplastic cells release signals which recruit mast cells and macrophages. So these are the macrophages we talked about before, and the mouse models have shown that if you eliminate MMP9, you don't get the response. You don't get this angiogenic switch here if you can knock out MMP9. And if you remove VEGF, you don't get the angiogenic switch. So this is just the experimental system, basically, by which people have made these diagrams. Okay, we know that because of this kind of mouse model. So this kind of experimental system where you can create this and then watch this happen in a different genetic background. by knocking out certain things. So you knock out VEGF and it doesn't happen. It tells you it's dependent on VEGF. It's like those experiments in the papers we see where you have a knockout of CREB and it still works. So CREB's nothing to do with it. You knock out PHF2, oh, now it doesn't work. You knock out VEGF, no angiogenic switch. Knock out MMP9, no angiogenic switch. Interfere with the signaling between the bone marrow or interfere with this recruitment event, no angiogenesis. Okay, so that's just to give you an idea of how you do these experiments. Last thing, going back to the therapeutic thing, it works well in mice. Here's a tumor that's been untreated. Here's a mouse that's had a tumor induced and surgically removed, highly vascularized. Exactly the same mouse model, treated with an inhibitor of angiogenesis. It works really well. This tumor is actually much smaller than this one. It's just been blown up, so they're both visible. But this one is much smaller than this one. So you can completely block angiogenesis. It doesn't work in humans therapeutically, though, for some reason. Don't know why. Okay, so that's heterotypic signaling and angiogenesis. The complicated part is the heterotypic part and the stromal support, and the angiogenesis part is the kind of more interesting thing. But you can't get to the angiogenesis part without doing the heterotypic signaling. So, okay. Next week, breast cancer case on Tuesday. All you got to do is bring yourself, and then exam on Thursday.